Well, good afternoon, and thanks for joining us for this May Dean's Research Seminar. I'd like to begin by acknowledging the traditional owners of the land I'm on today, which is the Wurundjeri people, as well as the traditional owners of the land that you're on, and acknowledge that Indigenous Australians have been custodians of the lands and the waterways of Australia for thousands of years, and I pay my respects to their elders, past, present, and emerging. Today, Professor Mark Stevenson will present his talk on unravelling population health problems in honeybees, koalas, domestic animals, and humans. Sounds pretty wide-ranging, but I'm sure it'll be absolutely fascinating. And he's going to talk about what you learn from one species that can be applied to others. Very much a One Health agenda. Mark holds a bachelor and master's in veterinary science from the University of Sydney and spent 10 years in private practice in New South Wales. He then did a PhD in veterinary epidemiology at Massey University in New Zealand, and thereafter spent another 17 years at Massey working in the veterinary epidemiology group before joining the University of Melbourne in 2014 to lead our veterinary epidemiology group. We're very pleased to have him here. Mark's special area of expertise includes quantitative epidemiology, spatial epidemiology, and infectious diseases modelling. And since joining the university, he's been awarded over 1.5 million of research funding as lead chief investigator, over 4 million as a collaborating investigator, and has produced over 160 peer-reviewed research publications on topics ranging from dairy cattle health and production to infectious diseases of livestock, wildlife, and honeybees, and he's completed 10 graduate researches. He currently supervises 6 PhD students and Masters of Science students. Mark's clearly a busy guy, and probably never busier than in the current epidemic when epidemiologists have been involved in so many different things. Please welcome Mark Stevenson. Mark, over to you.
Thanks for the introduction, John. Yeah, Mark Stevenson's my name. I'm a veterinarian and have worked in veterinary epidemiology, and I'm very proud to be the leader of a group of distinguished individuals who have helped to co-author this presentation, Simon Firestone, Anka Vietolter, and Caitlin Pfeiffer, all from the Melbourne Veterinary School. I'll start with just some background about epidemiology and veterinary epidemiology, and the focus of this talk will be about how veterinary epidemiology contributed to Victoria's COVID-19 response last year. So first of all, what's epidemiology? Textbook definition: it's a study of the distribution and determinants of health-related states in populations, and then the application of that knowledge to the control of health problems. And then if we think about how is veterinary epidemiology different, same definition, but we include issues related to production. So in veterinary epidemiology, we're not only concentrating on disease, but also on disorders of production, why some animals might produce less than others. Okay, and when you work in Building 400, a lot of lab work going on here, and a big difference between what the epidemiologists are doing with what the laboratory-based disciplines are doing. And I like to think in analogies, and of course the laboratory-based disciplines are looking down the microscope and looking for the fine detail of their particular subject area, whereas in epidemiology we're looking through a telescope and looking at the populations and making inferences about what we see by looking at population level health states and determinants of disease. And then to think about what's the difference between veterinary epidemiology and human epidemiology. So here I list a couple of key differences. First of all, obviously, we work with multiple species. Okay. Secondly, veterinary epidemiology, we have a lot more experience, and we're often called to deal with infectious disease problems. And this is a big contrast with human epidemiology. And if you think, the last time we had a large pandemic in a human population was in the early 20th century with the Spanish flu outbreak. Yeah. Number 3 is that animals are kept within groups, and this is what we call hierarchical data. So it's very common. We don't think of often animals at a single level. We're dealing with discrete populations, herds or flocks. And then finally, the veterinary epidemiology community is a lot smaller than the corresponding community of medical epidemiologists. And for that reason, there's a need for us to be across a broad range of technical areas. And this involves things like outbreak investigations, disease modelling, risk analysis and surveillance. And at the bottom of this slide, I cite a paper that Simon and I wrote back in 2015, not long after I arrived in Melbourne. And we talk about these differences between human epidemiology and veterinary epidemiology in more detail. Okay, so between March and December 2020, our group contributed to Victoria's COVID-19 outbreak response that was led by the Department of Health and Human Services here in Melbourne. And in this talk, I'm going to provide examples to show how the lessons that we've learned from previous investigations into the health of animal populations contributed to the COVID-19 response. So that's my aim. But first of all, to acknowledge the veterinarians that were working at Lonsdale Street at the Department of Health and Human Services. And we had a group photo back in June 2020, and here are all the veterinarians that were involved. We had about 12 in total, several missing in this photo. And those with a star next to their names are individuals that are associated with the Melbourne Veterinary School. So you can see all except 2, I think, of the veterinarians that were working on COVID-19 were related in some way to the Melbourne Veterinary School. And I think that's something we can be really, really, really proud of. And secondly, is that several individuals in this group, and Rebecca Gang, Jose Canavari, Philemon Hale, and Michael Moline have gone on into relatively senior roles within the department. And so that to me is a real, a very sincere recognition of the good work that veterinarians have played in this outbreak response. Okay, so I'll give 4 examples. So this will be a flying trip through animal and human epidemiology, and how we applied these examples to the COVID-19 response. And then right at the end, we'll come up with some general conclusions. So first of all, I'll talk about disease mapping. And switch over to animals now. Since the late 1990s, give you a little bit of history, there was a greater recognition of the importance of recording detailed information about farms and animal populations at risk. And this was largely driven by the epidemic of bovine spongiform encephalopathy in the UK, which started in 1986 and has continued. And I think the last couple of years, the UK has actually hasn't recorded any cases of BSE at all. So a very long outbreak control and eradication program, but one that has been largely successful. And in the early days of the BSE epidemic, it was very quickly realised that animal health authorities need to know where farms are located. And also, we need a way of permanently identifying animals. And early days, and I'm going back to the late 1990s now, we were able to take point maps of the locations of BSE positive farms. And this map on the left, we can get a general idea of the distribution of disease. But to make this more interpretable, useful for a decision maker, we can apply what we call kernel smoothing techniques. And this shows the density of BSE cases. This example, incident cases for the period 1992 to 1993. And we're using, of course, darker colour to represent higher densities of BSE cases. All right. So that's showing the distribution of disease. We can go one better than that. In epidemiology, we like to talk about incidence or prevalence, and calculating these measures, we think of a numerator and a denominator, and we can do exactly the same with our maps. On the top of our great big fraction here, we've got a kernel smooth map of BSE case farms, and then on the denominator of our fraction, we can have a kernel smooth map of all cattle farms in the UK. And what we can do is actually divide one by the other to produce an incidence map. And this is really useful for a decision maker because we can identify hotspots, cases where there's an excess of BSE cases after adjusting for the population at risk. And this is exactly the information that we need. Where are the high risk areas? Why are disease control efforts not working as well in these areas where they seem to be in others? Okay, so that's BSE in the UK. Now we'll switch over to a more recent example, and this is African swine fever in Vietnam. And Simon visited Vietnam in September 2019 and got out on farm and actually saw how the disease was affecting smallholder producers and how the disease was being managed. And what we can do, similar thing to what we did with the UK, we can map the distribution of African swine fever across Vietnam and provide some useful information on how the disease was spreading. Another example, we'll switch to Nepal now. And I'm going to talk about an organisation, an NGO called the Himalayan Animal Rescue Trust, otherwise known as HEART. And they've, for a good many years, been doing really good work on free roaming dog control in Nepal. And with HEART, HEART volunteers travel the streets of the largest cities in Nepal, capture dogs and desex those dogs, give them a vaccination for rabies, and then release them back into the community. Okay, and just last week, Caitlin Pfeiffer has been working with a DVM4 student, Sarah Tavlian, and this pair are doing some absolutely amazing work, taking data from HEART and mapping the distribution of dogs that HEART have been recording through their transect surveys, and producing some useful maps showing the distribution, in this case, the number of dogs per square kilometre in these urban areas. Okay, so three examples of kernel smoothing and how we can usefully apply that in animal health. Okay, now we'll switch over to COVID-19. And yeah, by way of routine in the Department of Health and Human Services, very common for diseases to be reported at what we call the local government area, or LGA area. And this is a fairly typical map that DHHS was producing throughout the last year's response. And what we've got here, this is for the week of the 9th to 15th of August, we've got the count of cases by local government area. Okay, now I'd argue for a decision maker, someone controlling an outbreak, this is probably not the best way to show the distribution of COVID-19 cases. And having a wide experience with kernel smoothing and working with point data to describe the distribution of animal disease, we took the COVID-19 cases and produced an incidence risk map, in many ways very similar to the map that we produced for BSE back in 2000. And as I say, for a decision maker, this provides a lot more detail of the specific areas of the city where COVID-19, where the incidence, the number of cases per 100,000 head of population was relatively high. And we're showing those areas using the contour lines. Okay, so now we can go one better than that. And now we'll switch over to New Zealand and we'll talk about honeybees. And in April of 2000, New Zealand detected a mite of honeybees called Varroa destructor. And this honeybee on the left-hand side of the screen here, you can see this large round disc on the back of the honeybee, and that's a Varroa destructor mite, okay? And this causes severe problems for honeybee health. In fact, Varroa has often been called the foot and mouth disease of honeybees. All right, so New Zealand had an incursion of Varroa. Australia, I think, is one of the last large countries in the world to be Varroa free. So we've managed to keep Varroa out of Australia so far. Okay, so switching over to Varroa in New Zealand at the moment, and first detection in the Auckland area, so the top of the North Island, and the Ministry for Primary Industries, once they detect a new disease, they carry out what's called a delimiting survey. And so all of the apiaries, we had our apiaries mapped fairly well in the Auckland area, and a transect survey was done to test for the presence of Varroa in apiaries. And in this map, we're showing Varroa negative and positive apiaries in the greater Auckland area. Okay, so the trick is now, how do we make sense of this noisy spatial pattern? And we do that, of course, using our kernel smoothing techniques. And we can do one better. We can overlay our kernel smooth map over Google Earth. Okay, so here the lighter colour is representing a higher density of Varroa positive apiaries. And I'm looking at that map and I'm thinking, my goodness, why are we having such a relatively high density of Varroa positive apiaries in this area of the city? Okay, go zoom further in. The great thing about tools like Google Earth is we can make this kernel smooth layer semi-transparent, and then I can zoom in further and to see what's underneath this high density, this high Varroa dense area. And wouldn't you know, it looks like Auckland International Airport. And so if I'm trying to understand how Varroa might have got into New Zealand, the high density around the airport would make you think that the disease came in through aircraft. All right, so that's using kernel smoothing and Google Earth. Let's switch over back now to COVID, yeah, and we'll take the map that we produced earlier, the kernel smooth map of Varroa incidence risk across Melbourne for August, and we can load that up into Google Earth. Now, this is really, really handy because what we can do is zoom into these hotspot areas to get a better understanding of what's happening in these specific areas. Now, I'm not showing it in this image, but what we did at the time is we loaded the point locations of households with COVID-19 positive cases. And really, really important for a decision maker to say, aha, this hotspot area here was perhaps due to 2 households where we had a high number of individuals, 6 or 7 people within that household that were COVID-19. And then we'd turn to the person who was managing those cases in that area and ask them, is everything under control? And if that was the case, then we could relax about that hotspot area. I'll just go back a slide so you can see. We could relax about these hotspot areas because we knew that the problem was being dealt with. Okay, so the value of the kernel smoothing, the value of using Google Earth, we could get the overall picture of what's happening with the disease. And secondly, with Google Earth, we could appreciate the distribution of disease in context of real-world geography. And that's really, really important for managing an outbreak response. Okay. Number 2, quantifying contagiousness. Okay. Now, home isolation is the cornerstone of the management of uncomplicated cases of COVID-19. So if you're diagnosed with COVID-19 and it's a relatively straightforward case, you're told to go home, rest up, and not to come out of your house for until you've recovered. So that's putting it very simply. Yeah. Now, a key question in the early days of the COVID-19 response was, is this strategy working? And under the assumption that exposure is occurring close to home, what we want to know is there an excessive COVID-19 cases close in space and time to identified case households. Okay, so if I had a household with COVID-19 patients, what I want to know is, am I more likely to see other COVID-19 cases close in space and close in time to those case households? And if I am seeing that, that tells me that something's going wrong with home isolation as a control measure. All right. So this is a really, really important thing for disease management during the COVID-19 to keep a handle on. We've got to know that this home isolation is working. And that boils down to a question of how do we quantify spatiotemporal interaction in disease risk? All right. Now, to do that, I'm going to quickly introduce you to a technique called the space-time K function. All right. Now, details, if you're looking for a reference, are provided on the bottom of the slide here. Yeah. But in a sense, what we do is we produce what we call a surface plot, and we have 2 axes to the surface plot. One is distance. The second is time, and on the 3 axes, we have a third axis in the vertical dimension, which is risk. And what we do is we quantify disease risk as a function of distance. Okay, so this is answering the question, what's the spatial component of space-time interaction? And then we can go back and we can say, okay, what's the temporal component of space-time interaction? Yeah. And then finally, we can colour this surface plot to show areas where there is an excess of cases attributable to space-time interaction. And this is really useful because it tells us how far this contagiousness is like, how far is that contagiousness extending in both space and time? And that's really important, yeah? Okay, now I'll flip this diagram around, and what I'm showing is the 0 point for time and space is shown at the back. And this, when I'm showing you a series of these plots, it just makes them easier to interpret. So I've showing you another D0 plot here. Yeah, again, the red area is showing the excess of space-time interaction. And now I'm going to go back to the UK, and we're going to talk about foot and mouth disease. And as many of you probably know, there was a large outbreak of foot and mouth disease in the UK in 2001. And huge problems, hugely expensive outbreak to control, and a lot of controversy at the time on the best way to manage the outbreak with depopulation, as shown here on the right, versus vaccination. Okay, so post 2001, we didn't do this actually during the course of the UK outbreak, but as part of the follow-up investigations after the outbreak, this was about in 2003, we applied this space-time K function analysis, these D0 plots to the UK 2001 foot-mouth disease data. Now, the outbreak went from February to September, and what we're doing here is we're producing a D0 plot for different phases of the outbreak. So I've divided the outbreak up into 4 periods. So So the first is from February to March, the early outbreak period. And you can see here that we really don't have a large amount of space-time interaction. Go on to the second period. Okay, space-time interaction is starting to kick in. Third period, May to July, and then fourth period, July to September. Okay, so the inference from this analysis is that space-time interaction, contagiousness, changes during the course of the epidemic. All right, now I'll take that experience we learnt from foot and mouth in 2001, and we'll apply it now to COVID-19 in Victoria in 2020. And now be aware that our human health colleagues hadn't used space-time K function at all, simply because they haven't had the need to deal with infectious disease. And so what we needed to do was, instead of throwing them a blue and red perspective plot, we needed to put that information in context with something that they were more familiar with. And that, of course, was an epidemic curve shown here up on the top left and a point map on the top right. And what we're doing here is we're taking time period slices. This is for the first wave of the Victorian outbreak. And we're computing for each of those time slices the D0 plot. All right, so the take-home inference from this was that, compared to foot and mouth disease, the amount of space-time interaction with COVID-19 was relatively small. The second inference is that spatial distance of contagiousness was relatively short. If there was a problem at all, it was more related to time. And that might probably relate to the length of differences in the length of time that individuals are remaining infectious. Okay, so similar to FMD, our D0's plots changed during the course of the epidemic. Point number 2, space-time interaction wasn't a feature of the early phase of each outbreak, foot and mouth disease, COVID. This interaction, this contagiousness, takes a while to kick in and to become important. And then third thing, particularly for COVID-19, keeping an eye on this quantifying contagiousness was really important for providing confidence in the effectiveness of home detention as a control measure. All right. Next, I'll talk about contact tracing. So this is a whirlwind tour through epidemiology and COVID. Again, go back to the outbreak of BSE in the UK in the late 1980s and 1990s. With the BSE outbreak, there was recognition of the need to not only record the identifiers of individual animals, okay, so not only to permanently identify them, but also to record when they move from one farm location to another. And in the mid-1990s to late 1990s, the UK introduced what we call the cattle passport system. Yeah, and this was every time an animal was moved, a record would be kept of that movement event. And since then, progressively, countries around the world that are exporting animals and animal product have introduced similar systems. And so they're now commonplace in the other European Union countries, Brazil, Uruguay, Australia with NLIS, Argentina and New Zealand. And here are some shots. On the left-hand side, I'm showing you the cattle passport system. Okay, so it literally was a physical passport. Now we've moved on to more electronic systems. On the right here, these are shots from 2003 and the similar system working in Argentina. Okay, so the question is, why go to all this trouble? Well, we go to this trouble because in the presence of an emergency animal disease response, if we've got these movement records, it expedites backward and forward tracing procedures. Okay, so a lot quicker and a lot easier to do tracing, which we need to do during an infectious animal disease outbreak response. And then the second reason for having traceability or a record of animal movements is that we need it for paddock to plate traceability in the event of a feed safety scare. And a typical example of that would be when animal feed becomes contaminated with chemicals such as dioxins. All right. A side effect of having a good animal movement recording system is that we can use a statistical technique called social network analysis to understand the connectivity between farms. And this slide, I'll show you just a very brief introduction or explanation of what we mean by social network analysis. We're representing farms as points, and every time a farm moves animals from one location to another, we connect those points using a line. And our question is, are the connections between farms at random? Probably not. Or do we have what we have on the right here is a network structure, which is very classic for what we call a small world properties. And so what we have is these little groups of farms that are all connected to each other and forming their own, what we call their own small worlds. Now, if we know that this property exists in a network, then it becomes a very easy job to stop the spread of disease from one small world to another by stopping the linkages between the small worlds. So this information is really useful for understanding our population of cattle and farms and the movements that go on between them. Yeah. Okay, so we can apply this information. Here's an example from Argentina. I'll just do this very quickly. Here's another example, a current project that we have working with the Ministry for Primary Industries in New Zealand. A report that we just delivered last week, literally, showing the movement of cattle across New Zealand. Caitlin Pfeiffer, for her PhD, did some really nice work looking at the movement of sheep in Victoria. And in Caitlin's work here, you can see these community structures being identified. And this is super useful information for disease control. Now, switch back to COVID. Okay. One of our ex-PhD students, Jose Canavari, had finished his PhD, was available, signed on with DHHS, has quickly moved into a senior role within DHHS, and talking with us during the early stages of the COVID-19 response. We're tracing questions about tracing and the need to carry out tracing. These issues were being raised. Very quickly, we realised that taking a social network analysis approach to contact tracing would be extremely useful. And so Jose led the development, working with a fantastic team of software developers to put together what we call the mystery case tracker. And this has really, really streamlined and made the tracing procedures within DHHS a lot more efficient. Yeah. So hats off to Jose for leading that work. To the point that in November 2020, there was a review into contact tracing across the different states of Australia. And we're very happy to say that Jose's, one of Jose's network plots made it to the front cover of the report. I don't know whether I should be proud of this photo or not, but yeah, great to see that work being usefully used. All right, to extend this idea a little bit further, genome sequencing often gets mentioned in the media when there's a hotspot of COVID-19. But to date, these ideas haven't been integrated with tracing tools with the mystery case tracker, and they really need to be. And in that respect, Simon's DECRA project called network modelling, nationally important emerging infectious diseases, is a natural and very logical extension to integrate genome sequencing into tools such as the mystery trace tracker. And so here are some shots from Simon's work that's come out of his DECRA project. And basically what Simon is doing in this project is reconstructing the transmission pathway between infected either farms or people in the case of COVID-19. And what he does to do that is using a combination of genome sequencing and also details of movement events in the case of animals or places visited in the case of humans. Okay, and so we really see that there's a need to integrate this substantial body of work into the COVID-19 tracing system. All right, 5 minutes to go. So next, I'll quickly talk about post-outbreak procedures. So we've largely got the outbreak under control. What's next? Yeah. I'll start with animals. Following an outbreak of foot and mouth disease in a non-endemic country such as Australia, we need to provide evidence of disease freedom to the international community before we can start trading again. Yeah, and what we do to achieve that is we carry out cross-sectional surveys, and where we go out and test and we look for disease and test animals for evidence of disease. And if all our tests are coming back negative, then we can make statements to the OIE that we've conducted so many thousands of tests and none of them returned a positive result. And on that basis, we can be 95% or 99% sure that if FMD is still here, then it's at a prevalence of less than X cases per 100,000. Okay, so that's quite routine work for mopping up after an infectious disease outbreak in animals. Yeah, and also given a portfolio of surveillance activities, the ways that we look for disease, and this could include slaughterhouse testing, testing individual animals on farm, and sometimes we test bulk milk. So we have a number of different things that we're using to provide evidence that we haven't got disease. Number two, we've got differences in the risk of disease across the different sectors of the population that we're dealing with. And then finally, we've got imperfections in the tests that we actually use to check for disease. So we've got three difficult things to deal with when we're carrying out this procedure. And yeah, given those conditions, how confident can we be that if all tests are negative, that we don't have disease? And then furthermore, with chronic conditions, if the search for disease is ongoing, and if we do one round of testing and find that all tests are negative, can we use that information when we come around in the next week to repeat our testing procedure? And this happens with diseases such as tuberculosis. So I do testing on one occasion for TB, 1 month, everything's negative. I come back in the next month and test again. They're all negative. Can I use those two pieces of information to build my overall confidence in disease freedom? Yeah, and that's what we use techniques, specifically scenario tree analyses. And this plot is very, very common. This example is for avian influenza. We're testing for avian influenza. I think this example was for wild birds. On each occasion, we're not finding disease. And over time, our confidence in disease freedom is increasing. All right. So this is really useful because it gives us an idea of when we've done enough testing. Okay, so Victoria, Australia, on the 15th of November, we had no COVID-19 cases for about 25 days. We're sitting there and asking ourselves, what's the strategy? Do we just keep testing until we run out of money? Or how can we quantify our progress? Can we use the same techniques that we use in animal health to the COVID situation? So, of course, you've got 12 veterinarians sitting in DHHS, and we think this is just like a post-foot-and-mouth disease outbreak situation. And we applied the scenario tree techniques. Yeah, and here I'm showing you a plot very similar to the avian influenza plot that I showed you earlier. Yeah, on the horizontal axis, we've got time, okay, starting from the 31st of October 2020 and going up as far as the 12th of December 2020. And all of our tests are coming back negative, okay? And on the vertical axis, we're showing our cumulative confidence that the prevalence of COVID in Victoria was less than 1 in 100,000, okay? And by about the end of November, we could be 95% certain that we'd achieve that target, yeah? Okay. Okay, so a useful way to put some numbers around and give us some guidance around the amount of testing that we need to carry out. All right, we can extend that work a little bit further. Ongoing work with colleagues from the University of Queensland, and I'll mention Jorg Henning and Ravi Disanyake here. Jorg and Ravi in Queensland have been using citizen science data to supplement transect surveys to estimate koala densities in southern Queensland. And relating that work to COVID-19, can we apply these citizen science procedures? And an important part of citizen science is a concept of search effort, how hard we're looking for koalas. Yeah, and of course with citizen science we have differences in search effort. We need to account for that when we're providing confidence that we've spotted all the koalas or estimating koala population density. Yeah. And so my question is can we apply these same principles, these search effort principles, to COVID-19 surveillance, particularly because testing is voluntary? Okay, we can't mandate people to be tested for COVID-19. Testing is voluntary. So I'm wondering if we can build in some of these techniques that we've learned for other species into what we're doing with COVID-19. All right, last point to make. COVID-19 vaccine hesitancy is an emerging issue of concern. And in many ways, the current issues related to hesitancy are similar to what happened amongst the horse owning population with Hendra virus. Okay, and here I'm citing some work carried out by Anchor and colleagues from the University of Sydney on horse owner attitudes to vaccination and ways that we can promote uptake of vaccination of the Hendra virus vaccine. And again, I'm asking the question, insights from this sort of work in Hendra virus, can we apply that into vaccine hesitancy in human populations? Okay, so some quick conclusions. In this talk, I've provided examples to show how prior experience with investigations of infectious disease in animals and veterinary epidemiology have provided, I think, a useful contribution to Victoria's COVID-19 response. My opinion is that, we've got good technical knowledge of COVID-19 control. It's well understood and it's well proven. I think the disastrous situation we were in Melbourne in August last year is testimony to that. So we've got the technical ability to control these outbreaks, but it is dependent on two things: strong leadership and societal compliance and discipline. And fortunately, in Australia, we're lucky enough to have strong leadership, particularly from the state premiers, and societal compliance and discipline. So big pat on the back to Melbourneites, especially for compliance and discipline. Yeah. Moving forward, the societal compliance in relation to vaccine uptake, I think, is our greatest threat in both the short to medium term, and that's both nationally and internationally. Okay, so I will draw a breath there, and I'll hand back to John.
Thank you, Mark. Maybe you can stop sharing your screen, and while you're doing that, I can say to everybody, can you please type any questions into the Q&A and not into the chat? And Mark and I'll pick them up, and Mark will try and answer the questions. Mark, maybe I can start, and I've got quite a few questions, but just to pick on one, how easy has it been to get data, the data that you epidemiologists need during this COVID outbreak? Because I'm assuming it comes from various different sources. And I'm thinking, for example, we've all been carrying around this COVID Safe app on our phones. Has that contributed to this, or does it have to come from the testing results that come from swabbing people's noses, or is it coming from the sewage? What have been the key sources of data that the you epidemiologists have had access to?
Yeah, DHHS has a fairly robust system for recording case event details. So I'll come back and I'll answer the question and think about numerator and denominator. Yeah, so the numerator are our cases, and DHHS have pretty good information for recording details of confirmed COVID-19 cases. Now that's details about the individual patients. So we've had privacy agreements and have had access to, for example, latitude and longitude of home addresses and those details. Now for the denominator, what we've used is the census data from Australian Bureau of Statistics, and that's recorded at a fairly detailed level of spatial resolution, a thing which we call mesh block, which is essentially a city block. And so we could use that as a denominator. So that allowed us to calculate incidence. We didn't use wastewater testing, and to be honest, the COVID app really wasn't the great success that it was planned to be. And I remember in the COVID-19 case details recorded by DHHS, there was a field for was this case detected using the COVID app, and I think someone will correct me, but I think the number was less than 20. Yeah. So yeah, unfortunately, it sounded like a great idea, and we had to give it a go. But the COVID-19 app didn't feature strongly in terms of case detection.
Okay, thanks, Mark. I'll let others come in with some questions. Let's see, Andrew Larkins, are you there? Would you like to ask your question? You there, Andrew?
Hi, Mark. Thanks for the talk. I was just wondering if you had much resistance from the human epis at all in applying vet epi methods that they might not have been familiar with.
I think they were generally amused by. We spent a lot of time in Lonsdale Street at the Department of Health and Human Services, and they were generally amused because us veterinarians would all sit and have lunch together and be talking about pigs and horses and all sorts of things. And often we'd be on the phone or on. Often I'd be teaching, so there'd be a few eyebrows being raised when I was talking about slaughtering pigs and African swine fever and different things. Was there resistance? A little bit of suspicion, but yeah, we already had quite good contacts, and this has largely come from Simon's relationships with DHHS, working with Q Fever. So we already knew the individuals involved, so it was pretty easy to get alongside them and say, hey, listen, this is what we do in animal health, and to show them different ways of doing things. And they were quite receptive and very happy. You know, the first to admit that, you know, this is the first time human health has managed infectious disease, you know, a large infectious disease outbreak in 100 years. So you guys coming in and talking about pigs and horses and honeybees and BSE, they were pretty happy about that. In fact, I think the most sincerest compliment I got was during a meeting one day when one of them turned around to us and said, "Jeepers, is there nothing you vets haven't had to deal with?" Yeah. So.
Okay. Yeah. Nice to see that interaction and that learning. Yeah. Robin Hall, you've got a question. You need to unmute, Robin. You have no microphone. Okay. All right. Then I will ask you a question, Robin. So Robin says, amazing presentation with some great examples. What are the considerations for spatiotemporal modelling when movement is long distance? For example, transport of animals over long distances or air flights over long distances, rather than direct transmission within a wild animal home range, for example.
Yeah, good question, Robin. In space, when we're modelling, particularly spatial modelling, we have to account for lack of independence in the data arising from things being close in space. And so we include the technical term as a random effect term to account for this closeness in space. Now that, and to do that, we define what we call a spatial adjacency matrix. And so if I'm sitting here and John's in the office next door, then we are defined as close in space. Okay, and that's taken account for in the model. Yeah. Now that there's some flexibility in the way that we can define that adjacency. And so adjacency might not be physical, but it might depend on travel time. And so if you, for example, the distance between Sydney, the degree of contact between Sydney and Melbourne, even though they're 900,000 kilometres apart, they are in fact quite close together because of the ease of travel and the amount of people travelling back and forth between Sydney and Melbourne each day. And so I think your question, Robin, is dealing with that long distance movement. That's probably how we would do it, by adjusting our spatial adjacency matrix in our models.
Mark, do you think that everybody was set up for this epidemic in the public health system? I remember when it was foot and mouth in 2001, and I was in the UK in 2001. One of the biggest problems everybody had was, well, after they'd worked out what the disease looked like, because they hadn't seen it for many years, one of the biggest problems they had was actually being able to record in a spatiotemporal way the actual cases. And they were faffing around for a long time until they worked out there were things like Excel spreadsheets. And, you know, it was just the basics that caused a lot of problems at the beginning.
Yeah. DHHS had very, very good systems for recording cases. So I don't think I could pull the Excel criticism out on DHHS. They had pretty good recording systems. The degree of familiarity with the staff with dealing with infectious disease, they not, you know, I don't think, well, there weren't very many staff at all that had dealt with an infectious disease outbreak. And so I think that's where, if we did anything useful, we could come in there as being 12 individuals that had actually seen infectious disease before and managed infectious disease in a population, albeit four-legged populations. And that was useful. So it was helpful, I think. Yeah.
Why do you think the spatiotemporal risk that you were talking about, why does that change over the course of the epidemic? Is it just high when there's a high density of cases?
I think there's a great deal of variation in compliance with disease control measures.
Oh,
yeah?
Okay.
And in the UK in 2001, you might remember Cumbria was an absolute disaster in terms of control, whereas the outbreak in Devon was all over by May. And so what we have very typically in outbreak responses is variation in compliance. And as a disease control manager, we need to understand that not everyone does exactly what they're told perfectly. Yeah, and so variation in compliance I think it's a given. You've got to assume it's some people are doing a perfect job and others are doing a not so perfect job. Yeah.
Okay. And I've got another question here from Robin Hall. Is the spatiotemporal change reflective of or correlated with the REFF, with the ref? I'm not quite sure what ref is myself, but maybe you'll know more.
Oh, yeah, R0, Robin? I think that's what you... the effective reproduction number, I think.
Yeah, the R effective, perhaps. Yes. Okay. Yes.
Yeah. Exactly. Yes, it is. Yeah, Robin. Yeah.
Yeah. R effective. Yeah. Okay. Mark, I think we're going to end it here because we're at the end of the hour, and we'd like to keep them on time. Thank you so much for that. I thought it was very gentle epidemiology, which was great, because I know it can all boil down to a lot of very complex equations, and I've seen those talks as well. So thank you for keeping it accessible to us all. And great work. Great to see you and your team and other veterinary epidemiologists working alongside the medical epidemiologists and contributing hugely to the analysis of what went on in COVID. So thank you for taking us through that.
Yeah. Okay. Well, I'll take that one on behalf of Simon, Caitlin, and Anka here. So backed up by a hugely supportive team. Yeah.
All right. Well, thank you to everyone who joined us for our seminar today, and I hope you'll be able to join us for our next seminar, which is going to be on the 8th of June, when we're going to have Professor Ian Clark and Dr. Surinder Chauhan, who will present on identifying heat resilience in farm animals. Okay. Thank you all very much, and thank you particularly to Mark. Bye-bye.
Thanks.